Hello everyone and welcome to One Academy. In this lecture I'll be starting with the mechanism of excretion. So I'll be talking about the first step in the mechanism of excretion, that is the glomerular filtration. Now glomerulus, as we already studied, is the tuft of capillaries, fine capillaries that are present inside the Bowman's capsule, and they are supplied the blood from the afferent arteriole, and then the blood is taken from them through the efferent arteriole. Now what all are the separations between the Bowman's capsule and the glomerulus? They are the endothelial covering of the blood vessels, basement membrane of the blood vessels, the basement membrane of visceral layer, and the visceral layer or the inner wall of the Bowman's capsule. So basically the cells of the Bowman's capsule. Now why are we actually why do we want to know about these all separations between the Bowman's capsule and the glomerulus? Because the glomerular filtration refers to passing of the filtrate from the blood into the Bowman's capsule. So when the filtrate is passing from the blood, it has to cover all this distance in order to reach the capsular space inside the Bowman's capsule. And as we already did in the structure of nephron, the visceral layer is facing the glomerulus, and anything or the filtrate that has to pass through the Bowman's capsule has to come through the visceral layer. Now what are the specific modifications in the visceral layer that provides a better function? That the visceral layer wall has the squamous epithelial cells, but in between these squamous epithelial cells are the podocytes. Podocytes have the evaginations called as pedicels, which are the foot-like evaginations, and these evaginations actually have the filtration slits. So the filtrate will pass through these slits and enter the capsular space. Now in the glomerulus also we studied that fenestration slits or the openings are present through which the filtrate passes from the blood into the capsular space. Now usually there are hormones, there are ions, and there are various other things that are passed from the blood into the tubule, the renal tubule, but much of it is reabsorbed back, because only the waste have to be excreted outside the body, other things have to be reabsorbed. For example, glucose, which is required by our body, is also a part of the filtrate, so it needs to be reabsorbed. If it is not getting reabsorbed, it shows that there is some problem in the normal functioning of the body. Usually when there is a very high intake of glucose, or where there is a disorder called as diabetes mellitus, when our body is not able to accumulate or accommodate the glucose, then it is excreted out of the body in the urine. And similarly the proteins are also a very important part, they also play a very important part in our body. So these are also not to be excreted out of our body, so they are also reabsorbed. Now first step is the glomerular filtration. How is it achieved? How is it that from the capillaries the filtrate or the substances present in the blood, they pass down into the Bowman's capsule? So before doing that, we need to cover the filtration pressure. That means what is the driving force for the passage of this filtrate from the glomerulus into the capsular space? So there are some positive pressures and the negative pressures, which finally sum up to give up the filtration pressure. First of all, the diameter of the afferent arterioles is greater as compared to that of efferent arterioles, so there is a pressure gradient that is created. Then we have the glomerular hydrostatic pressure. Inside the glomerulus is the blood flowing, and blood will have some hydrostatic pressure, as every fluid has. So the blood pressure in the glomeruli blood is called as the glomerular hydrostatic pressure, which is 60 mmHg. Now the blood colloidal osmotic pressure. There are different types of proteins that are present in the blood. For example, the albumin, the globulins, the fibrinogen, and etc. These provide a colloidal pressure of about 30 mmHg to the blood. Then is the capsular hydrostatic pressure. Now in the capsular space of the Bowman's capsule, there is some fluid that is present, and due to this fluid there will be a pressure which is called as a capsular hydrostatic pressure, which is again about 20 mm Hg. So this is the diagram which is going to show this. Here, from here it is the glomerular filtration pressure. Basically the difference between the two pressures constitute the glomerular filtration pressure. The glomerular hydrostatic pressure comes from the glomerulus and it is exerted on this capsule, the Bowman's capsule. Then the capsular hydrostatic pressure comes from the capsule, and this is the pressure gradient in between the two arterioles, because this one is larger in diameter and this is smaller in diameter. So what is ultrafiltration? Before that we will be studying the net filtration pressure, due to which the blood gets filtered, or due to which the filtrate passes out of the blood into the Bowman's capsule. Now the glomerular filtration pressure, that is the net filtration pressure, is equal to glomerular hydrostatic pressure, which is the positive pressure which allow the filtrate to pass from the blood, and the two negative pressures are the blood colloidal osmotic pressure and the capsular hydrostatic pressure. So these are the negative pressures which opposes the movement of filtrate, and the glomerular hydrostatic pressure is a positive pressure which allows. So the glomerular filtration pressure equals to GHP minus sum of VCOP and CHP, which comes out to be 10 mm Hg. So the filtrate components of the blood are passed out of the glomerular capillaries due to this 10 mm Hg of pressure. They pass through the endothelial fenestrations, the fenestration that are present in the capillaries, the basement membranes of the glomerulus as well as the visceral layer, and finally through the filtration slits of the podocytes to enter the lumen of the Bowman's capsule, or you call it as the capsular space. This process is the glomerular filtration, and the glomerular filtration rate is 125 ml of filtrate passes through the capsule per minute. So the rate of filtration in glomerulus is the filtration rate, which is 125 ml per minute. Now the filtrate consists of sodium ions, potassium ion, calcium, magnesium, phosphate, glucose, other hormones, vitamins, urea and uric acid. So why is this process called as ultrafiltration? Because there are very minute pores through which the filtration is occurring because of the application of a high pressure. That is why it is called as ultrafiltration. So this is all about the glomerular filtration. It requires a pressure to build up, and then there are small pores both in the capillaries as well in the Bowman's capsules visceral layer, which is because of the presence of podocytes that have pedicels in which the filtration slits are present. So these will help in the movement of filtrate. Apart from that, a pressure needs to be maintained which allows the movement of filtrate from the glomerulus to the Bowman's capsule. So I hope you like it. Thanks for watching.